Good evening and welcome to the 11th in our Webinar Wednesday series. Chag Sameach and Moadim L'Simcha, Shalom, Bonjour, and hello to all of you who have registered from around the globe. To be exact, Israel, England, the United States, and of course Canada. I would particularly like to welcome our very good friend David Levy, the Consul General of Israel to Eastern Canada. I am Mark Mendelson, the fiercely proud Chief Executive Officer of the Canadian Associates of Ben-Gurion University, and it is once again my pleasure and honor to be moderating today's program. I know that we have a competing television program in about 2 hours, and I can assure you we will be finished long before then, so please stay until the end, which should be an hour, 10 minutes. I am more than thrilled to tell you all that we have broken every previous Webinar Wednesday record until now. We have almost 220 participants registered for today's call. Of course, I'm sure that I don't have to remind you that the population of Canada, the Jewish population of Canada, is only 350,000, which just shows how urgent and relevant today's topic is and how curious people are to hear from Dr. Tal Zaks, medical school and PhD graduate of Ben-Gurion University, and certainly among the leading researchers in the world today, who is spearheading the race to the vaccine along with his research colleagues at Moderna. On behalf of the lay leadership and staff of CABGU, Canadian Associates of Ben-Gurion University, also known as CABGU, I welcome you and thank you all for joining us. I must say that this is just another feather in our Canadian cap of great successes during these difficult COVID times. As many of you know, less than a month ago, we had almost 800 guests buy tickets for our national virtual event featuring Mayim Bialik, President Danny Chaimovitz, and Senator Linda Frum. We also just completed a Support Our Students Emergency Aid campaign and successfully raised over $1.25 million in under 2 months. As I said earlier, I am definitely a very proud CEO. Please note that we are live streaming on Facebook. And just if you permit me in my position as CEO, I can take a liberty every so often. Just a personal shout out to my favorite uncle and aunt in London, England, both retired dental surgeons who are celebrating their 64th anniversary today and who are for their 8th or 9th time participating in our webinars. I love you dearly, Uncle Julian and Viorica. You are my idols. Our format today will be quite similar to our past webinars. For those of you who have attended any of them, I will first introduce Dr. Zaks, or Tal, who he has told me that I must call him, because I'm not his patient, thank God, who will speak for about 15 minutes or so, and then my colleague and Executive Director of Montreal and Ottawa, Simon Ben-Simon, will take over with the many questions we have already received from the participants and the questions which have already been sent to us now. We will then have a slight bullpen change today as Shimi Wenner, Executive Director of Toronto and the Atlantic Provinces, will be handling our technical issues instead of Zach Ostrov. Hopefully we won't have any Go Expos. Whoever has comments or questions, you will see the icon at the bottom of your screen, which you'll just have to click on to submit your question. Remember, the topic today is the race to the vaccine. Full speed ahead, or are we almost there? It is now my honor and pleasure to introduce Dr. Tal Zaks, Chief Medical Officer of Moderna Therapeutics, which was the first American company to enter a phase 3 stage of vaccine testing. As Chief Medical Officer, Tal Zaks oversees clinical development and regulatory affairs across Moderna. He received his MD and PhD from the Ben-Gurion University in Israel and conducted postdoc research at the U.S. National Institute of Health. He completed his clinical training in internal medicine at Temple University Hospital, followed by a fellowship in medical oncology at the University of Pennsylvania. Dr. Zaks, or Tal, began his industry career at GlaxoSmithKline in the genetics research group, where he built the oncology translational medicine team and led translational research on lapatinib, as well as the in-licensing and clinical development of fortinib. In addition to his industry work, Dr. Zaks is Associate Professor of Medicine at the University of Pennsylvania, has served as a volunteer physician at the Philadelphia Veterans Admin Medical Center, treating patients with genitourinary cancers. Prior to joining Moderna, Dr. Zaks was Senior Vice President and Head of Global Oncology at Sanofi, where he was responsible for all aspects of oncology drug discovery, development, and commercialization. Dr. Zaks serves on the board of directors of Adaptimmune Therapeutics, PLC. Tal, Dr. Zaks, the audience and computer screen is all yours. Take it away.
Thank you, Mark. I really appreciate that, the warm introduction and welcome, and I look forward to spending some time with you all this evening and to your questions. I'm going to, let me just make sure I can share my screen here. And let's put it on full screen. There we go. So I'm going to basically spend the time talking about how we are developing this technology, a messenger RNA vaccine. Before I start, I am warned by legal that I need to read you this large statement. Let me actually tell you what it is in plain English. This is actually a modern Wall Street interpretation of an old Talmudic saying that says that at the time of the Churban Bain Sheni, when we lost the Second Temple, since then prophecy has been given to fools. And so to the degree that I'm giving you any prophecies today, I am a fool. To the degree that you listen to me, well, that's on you. Who am I and how did I get here? Well, I actually started my career in medicine as a medic and medical instructor at the IDF School of Military Medicine. That allowed me actually to come to Ben-Gurion and really immerse myself in all aspects of both clinical care and academic medicine. And that was really a unique place for me, and I'm sure I'll come back to that at the end. But having been a medic, I now started my early years, the preclinical years. I worked as a nurse. I spent actually 4 years in the pediatric intensive care unit as a nurse. I then took some time off from the MD to pursue deep research in the field of immunology that had to do with oncology, how the immune system recognizes cancer. And then when I graduated, I spent a year of a rotating internship, and I had the opportunity to come to the NIH. Be'er-Sheva had set me up in two very important ways. The first is really deep and wide training spanning science and the clinic. And second, pretty good connections to the US. And I was fortunate enough to be able to come interview with Steve Rosenberg at the National Cancer Institute. He's truly one of the fathers of modern immuno-oncology, which is the only treatment form that actually offers curative intent treatment for people with metastatic solid tumors. After the NIH, I backtracked into clinical training, and at a certain point, once I came out of Penn as a fellow, I realized, okay, this has been fun. I've just spent 18 years in academic training myself, doing a whole bunch of this, that, and the other. It's time to do something in the world. And to the everlasting chagrin of my wife, who thought she married this good guy, I actually thought I had a lot more to offer in industry. And the reason was that in industry, which is where I spent the rest of my career, I realized that that's actually where drugs are made. And if I want to have an impact, well, I will have less papers to my name, but I will actually be working part of a team that's truly dedicated for the mission I care about. And so I volunteered as a physician, but actually left the academic medicine track to really focus on drug development in the industry. Now, I'm going to bring us into where we are today, which is a cancer vaccine. I'm sorry, we have a cancer vaccine, but today I'm talking about a COVID vaccine. And I'll spend just 2 minutes here on what it is we're trying to do here. This notion that protein is made based on our DNA, on our genes, but through an intermediary called messenger RNA is also called the central dogma of biology. You can Google it and you'll see every organism that is alive has messenger RNA. What we've done is figure out a way to turn messenger RNA into a new class of drugs and vaccines. And so if we can actually take this and teach a cell how to make the protein, instead of giving the cell the protein, or in this case, a bit of a virus that causes, that is a vaccine, we actually teach the body how to make the vaccine on its own. And so this is what our drug kind of looks like from a manufacturing standpoint. You start with a small snippet of a DNA template. You then synthesize in the manufacturing site this messenger RNA molecule using buffers and a whole bunch of enzymes, but it's all an enzymatic process that happens in water. So our reactors are only about 30 liters, not 30,000 liters like the biotech industry. And then you encapsulate it in a lipid so that you protect it so it doesn't get degraded, and voila, that's it. I make it sound simple. It actually is, once you've unraveled a few of the key elements here, it actually is relatively straightforward. And the important thing is it's reproducible. So this is, think of this as information. A messenger RNA can encode for this protein, that protein, the other protein, just by changing its code. So we now have a system where all I do is change the code, and that becomes a different vaccine or a different medicine. And so we've used that now to make a vaccine against this piece of a protein from SARS-CoV-2. And this is the spike protein. So the medicine, the vaccine starts on the left. It's this messenger RNA I showed you. It's in a lipid nanoparticle. You inject it basically with the oldest delivery mechanism known to man. It's a needle and a syringe. And you inject it into a muscle, and it gets into the immune system. Once it gets inside the cells, as you can see here, the messenger RNA gets into the cell protein-making machinery, and the protein starts to make, the cell starts to make that protein. Now, what we're making is that bit of the virus that the body needs to recognize in order to reject the virus. And it's what's called the spike protein. Now, this is not a mystery. We've known that it's the spike protein pretty much from day one, because SARS-CoV-2 is a close relative of SARS-CoV-1. And so we've had years to understand this kind of virus. And we know that that's the protein that the virus uses to attach to cells. So if you can teach the immune system, don't worry about the rest of the virus. You don't need to see the rest of the virus. All you need to know is this piece of protein. If you can recognize that and block it, then that virus can't get into the cells. And I won't belabor you with all the science that leads up to it, but suffice to say that in every species where you put an antibody like this into that mouse, into that monkey, and now we know into the humans, you can actually prevent viral replication and even ameliorate disease of somebody who's already been infected. In terms of what this platform does, this is actually not the first one we've done, nor even the most complicated one. In fact, this is the 9th out of 9 viruses that we've tried against which we've been able to get a robust immune response. It's a fairly young company and process, so we've been in the clinic now for a little over 5 years. Our leading program up until now was something called cytomegalovirus, all the way there to the right, a very complicated virus. I won't get into it. We're about to start the phase 3 next year, when in the beginning of this year, along comes SARS-CoV-2. And this is the development paradigm here. And we were able to get in the clinic very fast. And we were able to do that for 3 reasons. One, this technology can move very fast. Because it's a simple enzymatic process, as soon as that's based on information, as soon as you have the information, you can actually start to manufacture. So within 2 days of the virus sequence being uploaded by the Chinese on January 11th, the Chinese uploaded the sequence. On January 13th, our guys looked at it, figured out what was what, and put it into production. On January 14th, my wife wakes me up in the middle of the night. She's the smart one in the family and says, has somebody looked to see that you actually got the right sequence? What if there's a different sequence out there? So of course I immediately broke in cold sweat, started calling people, and that's okay. Actually, it wasn't just one sequence, it's dozens of sequences that have been uploaded, verified, and ever since then, we've been keeping an eye out to make sure that we've got the right sequence. Within 2 months of putting that into production, the phase 1 started, and this is a US NIH one, the National Institute of Health started the trial. And we had already had a very good collaboration with them. They had looked at this platform from the sidelines now for a number of years. They've been collaborating with us on other vaccines. And so, in fact, we were down meeting with Tony Fauci back in last September, thinking what would be a demonstration project to show that this technology can actually move quickly for a pandemic, because Tony and his team have been worried about a pandemic for years. And, you know, middle of January, this is no longer a drill. This is actually live fire. So we all mustered up, and they put this in the clinic in the beginning of March. That was the phase 1, just a few dozen people. I'll show you some initial results. We then started the phase 2 once we knew the phase 1 was safe. And the idea of the phase 2 is just to expand the safety so that you know you're safe to go to phase 3, because we'd envisioned phase 3 would be very large. And indeed, a couple months later, once we saw the safety from phase 2 that was okay, we said, okay, time to go to phase 3. This phase 3 trial is very large. It's 30,000 participants. It's done in collaboration with the NIH. And this is going to demonstrate that whether indeed vaccinating with this vaccine can indeed prevent COVID-19 disease. So the endpoint of the phase 1, phase 2 is basically safety, and let's see that I'm generating the right kind of immune response. The endpoint of phase 3 is prove to me that indeed you can prevent disease, and that what you understand about your safety profile is reasonable. We still have a lot of work to do. Even beyond that, we got to go show it works in kids. We got to make sure it's safe in pregnant women, immunocompromised people, et cetera, et cetera. But the essence of the program here has been this phase 3. Now, people ask, okay, you've moved so quickly, you've never moved so quickly, clearly you're cutting some corner. And the answer is, we've been able to move so quickly for 3 reasons. Number 1 is, this technology could get started quickly. Number 2, we could move quickly throughout because the FDA has been working with us lockstep, and things that usually take them weeks and months to review, they've been reviewing in days. And we've had a very close collaboration. We have a pretty robust civil service in this country, and indeed, they've been working nights and days just like we have to look over our shoulder and guide us as to where this needs to go. And number 3, usually phase 3 trials take a long time because we're waiting to events to happen. Unfortunately, we are living in the midst of a pandemic, which means that cases are occurring. The paradox of phase 3 trial in vaccines is the worse the pandemic is out there, the quicker we will actually have an answer of whether this is working. And so we've started the phase 3 at the end of July. We're going after areas that have very high transmission rates, and so we expect to see the answer quite quickly. The data, the initial data has been published in the leading medical journal. You know, if you're a business person, you read, there's only one journal, it's called the Wall Street Journal. If you're a medic, there's only one journal, it's called the New England Journal of Medicine. So our data have been published in the New England Journal of Medicine twice now, one for the young adults. This is data from the older adults 10 days ago. In fact, this data has been so cool that the New England Journal of Medicine has actually published our non-human primate data. And believe me, they don't often do that. All the data so far has been consistent. It's demonstrated that we can induce an immune response that's even higher than you can see in people who've been infected. And that's important because people who've been infected, generally speaking, there's only one case report that says otherwise. If somebody has been infected, they don't get sick a second time. And so the level of antibodies that they get are expected to be protective. And that's also consistent with all the work we've seen in animal species. We've also described the safety and tolerability. And when you think about vaccines, safety and tolerability mean 2 different things, and it's worth double clicking on. Tolerability is how tolerable is it? That is how much expected discomfort you're going to have. These are what's called solicited adverse events. That means we go out and asking for them because we expect them. If you're activating the immune system, you're going to get a little bit of flu-like symptoms. You get some fever, chills, fatigue, headaches. You're feeling yucky that evening, maybe a little bit the day after, and then it all goes away because the protein production goes away. Our vaccine, the messenger RNA, is a very transient molecule. It lasts for a day or 2 in the muscle, in the lymph node, and then goes away. It doesn't integrate with your DNA. This is not genetic engineering. This is all a very transient process. And so what you see is the expected reactogenicity. The proof of what I'm telling you is you can see all these bars are much longer on the right after the second vaccine than after the first, which means that all of these symptoms are happening more once your immune system is already recognizing the protein that it's making. Now, safety is something else. Safety means, well, how frequent do we see bad stuff that we don't anticipate, we maybe don't expect, but is worrisome because it is serious? And so far, we have not seen any serious adverse events that are related to our vaccine in these early phase trials. And of course, safety is something that we continue to look at and will continue to look at even after approval and after deployment into the population. That being said, there's no black box of mystery here. The messenger RNA that we make is similar to what is already endogenously in our cells. It is a fraction of 1% of the total amount of message that you can introduce into any given cell, and it's transient. The protein that we make, of course, is naturally produced by the body, but it is a foreign protein. It's the same protein that the virus would produce, more or less, if you were infected. Just to finish with a couple of words on our manufacturing, this is what our supply chain looks like. And it's important because while these require -20 to store and ship, at the end of the day, once it comes to a pharmacy near you, it goes into the freezer until it's ready to get used, and then you thaw it out. It's good for a week in room temperature. I'm sorry, in 2 to 8. During the pandemic, every vial will have enough for 10 people. We're trying to do the best that we can with what are limited supplies of actually glass and filling capabilities worldwide as we get ready to turn around the tide of this pandemic. And this is important because it underscores a lot of conversations we've had with various governments across the world. the US was the first that stepped in and supported the development, and of course later came with a large order to make sure that there is a significant amount of vaccine available for the population. I'm happy to say that both Israel and I believe Canada have disclosed that they've entered into agreements with us without disclosing the details to start to line up some access for a vaccine. Of course, we're a biotech company that's never yet sold a vaccine, so this is our first product, and that kind of support has been really crucial for our ability to advance our manufacturing at the same time as we're advancing the clinical trials. This is just to basically repeat what I said. It's pretty straightforward, doesn't require any new infrastructure for deployment. And so we're in the US already in deep discussions with the government to see how deployment will happen. The CDC is getting together its experts. The National Academy of Medicine has written a 140-page report on who should get it first. And ultimately, in the coming months, I think once, knock on wood, we will see that this vaccine is indeed safe and effective, the attention is going to shift to who should get it first, and ultimately, how do we deploy this out to millions of people. We're on record as saying we think we can make between 500 million and 1 billion doses next year. Our vaccine requires 2 doses, a prime and a boost, a month later, so divide that by half, and that's how many people we hope to protect from COVID-19 next year. That's really it at a very high level. I wanted to make sure that I kind of touch on the highlights and maybe give you enough to ask questions. And let me stop my soliloquy now and move it over to Q&A.
Thank you very much, Tal. I think that a lot of the presentation dealt with some of the questions that we've been receiving, but let me try to synthesize some of the most pressing issues and then have more of a discussion. So there's concern because of the politicization of the whole vaccine and the race of the vaccine, and in terms of being able to trust the vaccine that will be coming out through the United States or another source. And can you please elaborate on that? And one of our Canadian participants is telling us that he will only trust Health Canada's approval of the vaccine and is a little skeptical about the FDA's approval.
So I have seen nothing to make me personally skeptical of the FDA's approval, except to state that by some measures, they're being overly conservative. And what do I mean by that? Well, the Emergency Use Act was meant to approve a product when they think it may be effective. And they said they won't approve it until they know it's effective to the same level as required a full approval. There's an approval pathway that's called an accelerated approval, which I believe you will see some European countries potentially start to approve vaccines based on those endpoints. The FDA has said no accelerated approval, you will only get full approval, but we will consider an emergency use only when you've proven that this vaccine, beyond a shadow of a doubt, can indeed prevent from disease. Second point, we are in discussions with Health Canada, with the European EMA, etc. The other regulatory agencies that I've spoken to have not set a more stringent bar for approval than the FDA has, to my knowledge, to date. Okay, so take it for what it's worth. The final point I'd say is, unfortunately, this has gotten politicized to the point that nobody wants to have a discussion on the data and the content, but people just assume if Trump okayed it, it must be bad, and vice versa. If it's good, let's not let Trump okay it, and let's wait. And I would rather we move the conversation to understand, and hopefully we will in a very public debate, what are the data? Why is the FDA taking the stance that it does? And the FDA will hold a very public hearing on this. And my goal is to make sure everybody understands the data and makes their own decision. The final point I'd say is, look, you know, everybody worries about vaccine hesitancy and what happens if people don't take it. I'm going to give you a very Israeli answer. If you don't want it, don't take it. I'll tell you why. we're going to have demand outstripping supply, certainly through the first half of the year. And I can tell you that those who are hesitant are going to stay at home and at risk. And those who will take it based on the science will have a much higher likelihood of protection. And I expect that anytime there's a sense of supply and demand and there's not enough to go around, and there's enough people who believe that what's going is actually good, you're going to see public sentiment flip really quickly.
Okay, thank you. Well, I think we all feel better. So let's elaborate a little bit in terms of the discussion around when will there be enough dosage for an international vaccination program so that people can go back to a normal life of being able to travel everywhere where there's not going to be a gap between the richer countries versus the developing world, where what is the plan to have an international program of vaccination?
I wish I had an answer for you. I don't. I think the truth is that there are a lot of good-intentioned people, primarily in Europe, that are trying to see how do we get the entire world vaccinated. But the truth of the matter is that every government's first charge is to take care of its own citizens first. And I think what you're seeing is, you know, call it realpolitik or whatever. But I think the reality on the ground is that every government, first and foremost, is trying to take care of its own citizens. And I think what you will see deploying, and it's an answer to one of the questions up there in terms of 12 months away versus months away, I think it's months away from the first data and first doses being deployed to those who need them the most in the United States. I think that over 2021, you're going to see a ramp up of production such that if you tally up all the companies that say they will have enough, and you assume that all the companies that say they will have enough will actually have vaccines that work, we should probably be with several billion doses available for humanity by the end of next year. Now, whether all of them work to the same extent and whether all of them can be deployed to the same extent, and who will pay for all of them, a lot of that still needs to get worked out, and I wish I was wiser than I am today. I'm not. My focus has been to make sure that we are demonstrating that what we have has the potential that we show is proven to prevent disease, and then scale up to the best of our ability, financially, scientifically, and politically, to supply as many people who need it across the world as we can.
Is there any kind of coordination between the various pharmaceuticals and companies such as yours to coordinate such an effort?
No, I think that would be violating antitrust. I think in the context of a pandemic, what's happening is that governments are pulling companies together. So I sit with other of my colleagues from other pharma companies who are being funded by the US taxpayers. We sit meeting together and discuss how do we work together with the US government, how do we harmonize what we're doing. But it's only the US government, by virtue of bringing money to the table, that's actually brought companies to the table. I think what you're seeing in Europe is there are organizations that are trying to set up a big pot of money and then invite companies to kind of put their wares up and figure out what the right price point is and when they will have available supplies. I think a lot of the richer world will probably have access through 2021 to the question on travel. I think getting to the nth billion person on the planet will probably take us beyond 2021, the way I currently see it. And I assume that's going to have an impact on the way life returns to normal or not. That being said, once you have herd immunity, then anybody who's been vaccinated, for them, life in their local area should probably be returning to normal. But again, I reference my first slide of the deck in terms of prophecy.
Great. Okay, let's turn a little bit to some more scientific or related questions. And the one from Dr. Stephen Klein in Montreal, actually. Do you believe that the responsiveness of the FDA for the various COVID vaccines, small molecules, will translate down the road to other indications such as oncology, i.e. short path?
FDA has been pretty good with oncology. I happen to be a medical oncologist my entire career who stumbled into vaccine in the past 5-6 years. It's the oncology division that's actually set the path. And by the way, as an aside, Peter Marks, who's one of the best civil servants I could ever hope to work with, who's running that division at FDA, happens to be a medical oncologist by training and by what he did most of his life. So I do think there is an unmet need mindset that oncologists have brought into both drug development and drug approval that you're seeing play out here. And I've no doubt will have repercussions for the future as well.
Great. A question about how are trial participants exposed to the virus?
They're not.
Okay.
Trial participants are asked to go about their lives as they would if they were not on trial. We have selected trial participants who are at greater than average risk by virtue of occupation. So not people like me who can do their work remotely sitting in their living room, but people who have to be out and about in the world. But basically we're not exposing anybody. Purposefully, we're asking everybody to be careful. We know that by chance, unfortunately, some of them are still going to get infected.
Great. Okay. There's a question from Mr. Alexander Gorin in the United States. There are endless rumors about COVID-19. One is that it may cause permanent damage to some organs. Assuming that the vaccine is successful, would it prevent permanent damage to some organs?
Well, to the degree that the virus causes long-lasting effects, then preventing somebody from getting sick and having a significant infection, then I would expect the answer is yes.
Good, thank you. There's an interesting question. You mentioned that the vaccine would need to be injected twice. Can you elaborate on that?
Yeah, one of the things that's true fundamentally of our immune system is that if you come and you vaccinate somebody one time and then that protein goes away, well, the immune system generates some antibodies, but then everything's quiet on the Western Front, so the immune system goes to sleep. If you come back a month later and you say, well, hold on a second, that thing you thought you got rid of, you actually didn't, you kick the immune system into high gear because, okay, now I have to go do something better. Clearly this enemy ain't gone. And so when you can do a prime and a boost, what you get at the boost is usually much higher levels of an immune response and better memory against this thing. And so that's just general immunology principles. Now, not every platform of vaccination can actually come back a month or two later. Some of them, if you're using a whole virus and this protein is just a smidgen of it, like the adenovectors, it's really hard to do to come back a month later and give a second boost because now the immune system sees all the rest of it and doesn't allow you actually to express the protein you care about. And so one of the good things about our technology is that you're actually able to come back and boost that immune response. And so make sure that we have a higher ability to prevent infection and likely a longer durability of effect. Although on the question of durability, I think it's going to take us months down the road to figure that out.
Okay, there's actually two related questions. Will they need, what do you expect the immunity to last? And will we need an annual booster shot?
So that's a big unknown. Will we need an annual booster shot? Personally, for most of us, I think the answer is no. Remember, the reason we get an annual shot for flu is not that last year's shot doesn't work. It's that this year's virus is different. If you were to see last year's virus again this year, you'd still be immune. In fact, most vaccines, you take Pneumovax, even in the elderly, you don't give it a second time, maybe 5 years later if you're immunocompromised. And most vaccines don't require an annual, in fact, I know of no vaccine that requires an annual boost. So I think that in the first year or two, for those of us who are old or immunocompromised or are at risk, especially if we got a vaccine that maybe wasn't that effective to begin with or our antibody levels have waned, I think there's going to be a question of, should we just take a boost just to be sure? And I think that's valid, although it's going to be hard to prove. But I think in the long run, unless the virus mutates like flu to something else, the immune system should probably last for a few years if it works to begin with.
Do we know if the virus mutates and what is expected?
Not yet. 9 months in, we've seen several mutations. In fact, the mutation that's most broadly circulating now is different than the one that was originally described at Wuhan in one snippet. However, those are mutations that enable the virus to spread better, but not because they are immunological resistant. So it's not an immune escape variant, and that's important. If we take blood from somebody who's been vaccinated with our vaccine that's against the original strain, and we put it against a virus that's this new virus, it works just as well. If anything, it works a tad better. So none of the mutations we've seen to date that are circulating to any significant level are actually ones against which we think the vaccine isn't going to work. Now, if that changes in the future, we're going to have to adapt our vaccine and change the code of our vaccine. That's the beauty of having such a digital, flexible technology. We'll just change the code to match the new mutation, and we'll start making a new vaccine.
There's a number of questions about time frame. Really, people really want to get back to their lives and to be able to, to just get back to normal. This new normal is not working for most people. And can you elaborate a little bit? Dr. Fauci on CNN a few nights ago mentioned that he expects a vaccine by January, February that will be rolled out to certain segments of the population, medical personnel at the beginning, and then seniors, and that by within a year there will be mass vaccination. Do you agree with that assessment?
Yeah, I think he's right. I think he's right on two counts. First, I think in the beginning, everybody will want, in any country, will want to deploy it first to first-line responders and those at risk for serious disease. He's right on the second count. If you look at the total quantities being lined up by different manufacturers, I think they come online with tens of millions of doses, really, and then hundreds of millions of doses in the first and second quarter of 2021. My sense, personally, is that the coming months are still going to be tough. I think this winter we're still going to have to brace a little bit longer, but I hope that by the end of the winter, spring, we start to vaccinate people in significant numbers through the next summer, such that by the time the next school year rolls around, by and large, life can be back to normal. That's my hope.
Thank you. There's a business-related question. Will you license manufacturing to other companies?
So far, we have not contemplated that. We don't think there's much utility in doing that because we've simply contracted, we've expanded our capability by contracting what are called CMOs, contracting manufacturing organizations. So we have a strategic collaboration with Lonza that has a facility in Switzerland. And so we're enabling to leverage other companies, not by licensing our product to them, but by actually collaborating with them and having them produce it under our own processes.
There's a question. What about the data indicating that the level of antibodies reduces over time in people who have been exposed? And that leads to another question, which asks about, will there be the ultimate protection or the ultimate vaccine? Will that include several types of vaccines so that there's full coverage?
Well, almost all the vaccines that I'm aware of, and certainly all of the ones that are in the front line, are using the same antigen, the same bit of virus, that same spike protein. So it doesn't make sense to combine different vaccines in one. It makes more sense if you have many vaccines available to go and immunize more people. The question of waning immunity is a good one. I will make 2 points on that, 3. One, if you look at the data, it's controversial. There have been some small studies suggesting waning immunity. There have been others that actually, there was a New Zealand large trial recently reported that actually demonstrated that they hold on quite significantly. Second point, it seems that the sicker you are, the more antibodies you have and the longer they stay. And I think that's consistent with the clinical data that if you get sick, you're not going to get sick a second time. Now, you may get infected a second time, which means transiently somebody may be able to detect by PCR that you've got the virus again in your nose, but by and large, you're not going to get clinical symptoms from that. And since our vaccine induces antibodies at levels that are even higher than those that on average you see in people who've actually gotten clinically sick, we believe that this vaccine should have both the potency and the durability to protect people at least to the same level. Now, the last piece is, let's assume worst case scenario that immunity wanes, and if all we know about this history so far is that it lasts for 9 months, because so far people are not getting sick a second time. So let's say it only lasts for a year and then you're at risk again. Well, what's the alternative? We still have to get this vaccine out. We still have to give it to everybody. We'll worry in 2022, 2023, whether people need a booster shot or not. I don't think it changes anything we really do in the next year or so.
Okay, so that's an interesting point, because there's another question about how will you keep up with the changes and its possible mutations of the vaccine?
Oh, I've got a leg up on that one. That's easy. You've seen us because we've got a digital information-based vaccine. Once we prove in principle that it works the first time with a phase 3 trial, what we would do is similar like flu. You don't run a phase 3 trial for flu every year. You just take a proven technology, you put in a new sequence. Now, you've seen us be able to go in 2 months from picking a sequence to dosing people. That was in a clinical trial, but once we have a manufacturing footprint that's able to produce 100 million doses a month or what have you, then you simply change the sequence and you start producing a new vaccine. And so if we need to amend our vaccine because there's a new sequence, we'll probably end up calling it SARS-CoV-3, and we'll have a SARS-CoV-3 vaccine, but the route to that vaccine is going to be much, much quicker for the reasons I mentioned than what we're currently facing.
And someone's asking, what makes your vaccine, the Moderna vaccine, safer than others, or is it safer than other vaccines being developed?
Don't know yet if it's safer. On the fundamental principles, though, because the body is producing the protein on its own, what we're producing in our body is a naturally occurring protein. So to the degree that there are any safety elements that are brought in because you made the wrong protein, we don't have that risk. We're not considered an adjuvanted platform. So to the degree people worry about these non-specific immune stimulants as having safety side effects, we don't have that problem as well. We do have a lipid nanoparticle, and that's a little bit new, so we're going to have to watch out for that. But that's a very rapidly biodegradable lipid, so I'm not expecting anything untoward from that. Now, so anything that's specific to our platform is not keeping me up at night. There may be cross-reactivities that, once in a million, because of the spike protein, you see the immune response mounting the wrong kind of response. If that happens, I think that's going to be something that's common to different vaccines. Those of you who've heard about the case for AstraZeneca's vaccine, they had a safety event that put them on hold, and they're still on hold in the US. Well, I can tell you, and this is not me telling you, that my Data Safety Monitoring Committee is an independent group of experts appointed by the US NIH. They look at our data every couple of weeks. They haven't come back and asked me to do anything differently and haven't told me I've got a problem. And the FDA, and they are the ones who are actually looking at AstraZeneca's trial as well, because it's the same Data Safety Committee that the NIH has appointed to look at all the sponsors. And the FDA hasn't come back and told me to do anything different on my trial based on AstraZeneca's finding. So far, I think the scientific world and the regulatory world, at least, are differentiating and are not having the same concern around that particular safety event with our platform, or frankly with Pfizer's, which is using a very similar platform.
So you're right that Canada has signed a deal with Moderna to buy vaccines. And do you have a cost of what will the vaccine cost?
So the costs that we have negotiated with governments are a function of the volume writ large, and the cost per dose has been roughly somewhere between $20 and $37 a dose, depending on how big the order is. Obviously, the US government had the biggest order, and in these days and this current environment, as you would anticipate, the US is not going to foot the bill and pay the highest price relative to any other country.
So you've mentioned, there's a kind of cute question, you've mentioned what doesn't keep you up at night a few times. And one of our participants wants to know, what does keep you up at night? Professionally.
I think the thing that keeps me up at night is the ability to explain our science and our findings at a time to a public that is highly fractured in its in how it gets its communication during an era where our communication venues have all been polarized. It is very hard to have a dialogue on the content, and there's no more sort of, the old ways of communicating are sort of gone. And that worries me, because I'm on the cusp, I believe, of one of the greatest achievements in modern medicine and modern science. And for somebody who's lived their entire life with the mission of translating science into medicine, I'm on the cusp of what I think is the most impactful finding, and I find myself showing up and getting a very strange response from the world, half of it saying, yeah, that's great, more, you're not moving fast enough, and the other half saying, well, I don't believe you. And a communication atmosphere, an environment that has lost that middle path of just having a common sense discussion.
Absolutely. It's quite amazing. Well, thank you. I think there are questions about the vaccine for immunocompromised people and the risk around people who are immunocompromised around the vaccine. Can you elaborate a little bit on that?
Yeah, I care about that deeply as an oncologist, of course. I think the risk is that the vaccine doesn't work well enough if your immune system isn't able to mount a response. I don't think there's any excess risk from a safety or tolerability perspective. In fact, if anything, the opposite. I worry about, the vaccine taking, as we used to say in the old days. That being said, if you look at the history of vaccinating cancer patients and other patients who are immunocompromised, by and large, unless you are really under acute and severe immunosuppression, we're able to vaccinate our cancer patients just as well as we are able to vaccinate most people. But we need to prove it. So we actually are talking to the National Cancer Institute in the US to see if we can start a study as soon as possible to prove the point that indeed this vaccine will work. I can tell you as an aside, I know the platform works for cancer patients in terms of mounting an immune response, because one of the coolest things we're doing is actually a cancer vaccine. We're taking cancer patients, we're making a vaccine just in time, just for that one person based on their cancer's code. And we're trying to fit what the vaccine needs to do to wake up that individual's immune system. We've been at it now for about 4 years. We're in a randomized phase 2 trial. And what we've seen is that we can actually induce an immune response in those cancer patients. So I know the platform can work in cancer patients. We need to prove it in this application.
Great, thank you. A few more questions. This will be the first mRNA vaccine. Does that concern you, as there are no others to compare?
Does it concern me?
Does it keep you up at night?
Not, again, only from a public perception. It's another, if you will, it's another talking point for the anti-vaxxers to say, oh my God, this is something the world has never seen. The mRNA is genetic material. What are you doing? When the science is actually not, there's nothing big or magical about this, and this is not genetic engineering in any sense. And so I worry that the first application that we have, people will use the fact that it's new in an unscientific manner to generate anxiety and fear where one shouldn't exist. So it would have been easier, I think, for the public at large if this would have been our third or fourth vaccine. On the flip side, the next vaccine we make against, to prevent cytomegalovirus in newborn infants, which is the #1 need in terms of congenital birth defects, at least the overhang will be gone because everybody will have seen an mRNA vaccine given to 100 million people. And so, I think there's also a silver lining there.
So there's another, people want your opinion on the way that different countries, states, provinces, cities have either shut down, reopened, and a number of people want to know your opinion about reopening schools, for example, and should we be sending our kids to school, or?
I'm the wrong person to ask about that. I have no particular experience or expertise that would make me an expert more than the man on the street.
Okay, or more than a parent sending their kids to school.
Exactly.
One last question, and I'll use my BGU hat. Nowhere did I read in your CV that you're actually from Beersheba, but and there's some excellent medical schools in Israel that you alluded a little bit to why you chose Ben-Gurion University's medical program, but can you elaborate a little bit on how you ended up at BGU and Ben-Gurion University and Beersheba?
Yeah, true story. So when I left the army, I was actually accepted into every one of Israel's 4 medical schools, some of them with full scholarship. The only one that put me on the waiting list was Beersheba. However, my girlfriend at the time had the opposite experience, and Beersheba was wonderful enough in offering both of us a spot and getting convinced that me, even though I wasn't the kind of fit they were necessarily looking for, because it was more about community, and I was, you know, even as a younger man, I had a passion for the sciences. They decided to take a chance on me and accept me nonetheless. From my perspective, you know, putting the personal part aside, I very purposefully sought an environment where, and I remember Dr. Tarek was her name. She was a wonderful community physician. Her husband was an orthopedic surgeon, the head of the department at the time. And I remember talking to her and telling her, look, I get the sciences. I'm not very good at sort of the communication piece and the patient outreach piece. And, you know, I was quite an introvert in those days. And coming to a place like Ben-Gurion was actually going to force me out of my shell in a way, and out of my skin and out of my comfort zone. And even then I had a sense that if I wanted to flourish across the spectrum of clinical through basic science, I really needed to get challenged on the domains where I'm uncomfortable. And so for me, I very quickly found Beersheba to be an ideal place. I actually had to twist the, you know, Shraga Segal's arm almost to take a year off. It didn't have an MD PhD path at the time, although since then they have a path, and I recently spoken to the MD PhD students in Beersheba. But taking a year off to go do deep science was not something that people commonly do. There was one of us every 2 years or so. And so I was able to leverage that unique pool, if you will, of Ben-Gurion to really start my journey of translating science into medicine by having a very solid foot in both camps through that unique environment of BGU.
Great, thank you very much. It was a Tal, and I'll take the liberty to call you Tal. It was an honor and a privilege, and it was an absolute delight to speak to you and have you on, and I thank you, and I'll turn it over to Mark for the last few words.
Okay. Simon, you've got to let me go. Am I here?
Yeah, you're on, Mark.
Well, one last question, a very personal one. Our executive director in Winnipeg, the youngest of our team, his wife is pregnant. And the question he's asking, if someone who's pregnant contracts COVID, can it be passed on to the fetus?
I don't think we know enough yet, to be honest. I certainly don't know the answer, but I expect that the OBGYNs there would probably be wiser than I am. I can, if they want to follow up with me afterwards, if they need some info, I've got a terrific, Alison August is one of the smartest, warmest OBGYNs I've ever met, who happens to be a physician, a senior physician on my team, except Saturday mornings when she talks to the rabbi and I'm not allowed to call her. So I'm happy to pass on the reference, and she would have a better answer than I would.
Well, all I can say that I was watching the Q&A numbers start at 1, hit 35, and then I know that there were at least 8 questions which were submitted beforehand. So altogether we had 44, 45, 46 questions, which is another record for us. Unbelievable. And I just have to thank you, say a huge toda rabba, rav todot. You are not only obviously a brilliant researcher, but you speak English flawlessly. There is not a trace of Israeli accent, and I'm married to an Israeli. I lived in Israel. Our kids are Israeli. So you appeal to everyone. And I think your handling of the questions and the warmth of your answers just allow me to say on behalf of everyone who participated that we are crossing our fingers so that you and your Moderna team will be the first ones out of the gate with the vaccine, and may you all win Nobel Prize for Medicine, which I'm sure will be welcome and deserving. Is all I mean.
Thank you for that. It's been a true pleasure and honor to be here with everyone, and thank you for having me.
Okay, I just have a few more minutes, so don't leave us. Although you're just going to have to pretend to be polite and listen. You know, I was doing some pre-research, and it was my understanding that there were over 160 what you call in the Branja, as they say, vaccine candidates, as they are called in the field, with 30 of them having already started their human trials. But you guys, as I said before, were the first ones in the States to enter phase 3. I also read that your late father was a mathematician, and my late father was a physician, a graduate of McGill of 1940. So we come from similar backgrounds of curiosity and science, and I always wanted to be a doctor until my second year at McGill, when I realized you got to study too hard. And thank God you studied and you became what you became. Very, very proud of you being a BGU graduate, because I have come to love BGU. And of course, this has probably become another should-have moment in my life, when I should have bought Microsoft 25 years ago and Zoom. I should have bought Moderna a few months ago. But, you know, what can I tell you? Anyways, a huge thank you to our listening audience for once again supporting us and the outstanding Ben-Gurion University. I would like to really thank my outstanding, excellent staff team across Canada, Simon Ben-Simon, who handled the questions with great aplomb and professionalism, Michelle Richmond, Shimi Wenner, Sam Cordes, Paulina Pshanishny, Zach Ostrov, and David Burson, another graduate from BGU. National President Mitchell Olbaum, his question about cost of the vaccine, a great mentor, advisor, and friend, and his leadership team. Thanks as well to my excellent colleagues in Israel, President Danny Shamovich, Vice President Or Chaimovitz, Vice President Jeff Kay, and his team, and Doug Sesserman, my colleague and friend, CEO of the American Associates. Please, everyone, stay tuned for our next webinar on October the 28th, which will be featuring Professor Nadav Davidovich, who is on Israeli news at least once, twice, 3 times a week, and Professor Daniel Filk, who will be speaking on pandemic in Israel. What the heck went wrong? On Sunday, October the 18th at 2 p.m. Eastern and 11 a.m. Pacific, the Jewish Federation of Greater Vancouver, in partnership with Canadian Associates of Ben-Gurion University, will host a webinar in Hebrew on the future of the tourism and hospitality industry in Israel post-COVID-19. The webinar, moderated by our own David Burson, Regional Director, will feature 3 BGU professors, Nadav Davidovich, Yaniv Poria, and Dean of BGU School of Hotel Management and Tourism in Eilat, all together with Dr. Amir Shani. We will be featuring on November the 18th, Professor Yuval Elovich, who is considered right now one of the top cybersecurity experts in the world, and who heads BGU's department, as well as a National Emergency Cyber Response Team of Israel, situated in our campus. Please look for your email about it. It should be another outstanding webinar. Again, thank you so much, Tal. Check us all out at www.bengurion.ca. Chag Sameach to all. Mo'adim l'simcha. Stay safe and wear masks. Toda rabah, Tal. Thank you so much.
Shalom, toda lekulam. Bye-bye.